Hi everybody, this is Noah. I'm one of your SIs for this quarter. So we've been talking about the central dogma. Specifically, today what we'll be going over is transcription. And so first off, let's talk about where transcription occurs. In prokaryotic cells, transcription occurs in the cytoplasm, while in eukaryotic cells, it occurs in the nucleus. So if let's take a second to think about why this is. So we know prokaryotes don't have any membrane-bound organelles. Because of this, their DNA is found in the cytoplasm, so transcription can also occur in the cytoplasm. Contrarily, if we look at in a eukaryotic cell, the DNA is found specifically in the nucleus and it can't leave the nucleus. Because of this, transcription has to occur in the nucleus where that DNA is. So there's 3 steps to transcription. The first one is initiation. And so we have our RNA polymers, which will go ahead and bind to the promoter region of our DNA strand. And if we take a look at our double-stranded DNA, we'll see over here that we're going to be replicating based off this bottom strand. And so because of that, we can consider this bottom strand the template strand because we're using it as a template to form our new RNA molecule. And because of this new RNA molecule will be complementary to this template strand, meaning that it'll match the sequence of this top strand. So we can call this top strand the coding strand because the sequence of this top strand will match the sequence of that new mRNA that's formed. The second step of transcription is elongation. And so we have our RNA polymers that's going to make its way down the DNA, and specifically in the 5' to 3' direction. And so this new RNA molecule is going to be formed in that 5' to 3' direction as it moves along, copying from that template strand. Last step of transcription is termination. So here, eventually our RNA polymers is going to make its way to a stop codon. And once it reaches that stop codon, the newly formed RNA will go ahead and be released. Along with that, RNA polymers will go ahead and detach itself from the DNA. So now we'll have that newly formed RNA molecule, and our DNA will go ahead and rewind itself up. And now let's talk about transcription factors really quick. So transcription factors are proteins that bind to the promoter region, and these guys help regulate which genes get expressed. And so we can have this TAT box. Transcription factors will go ahead and bind to that and form our transcription initiation complex that will help our RNA polymers to bind to the DNA and allow transcription to occur. And so really quick, here's a little summary page of everything that we talked about in this video. If you guys have any questions, feel free to reach out to any of the SIs. We're always here to help you guys. And if you have any questions, my email will be in the description. Thanks, guys.